I'm oral historian Mike Chappell, and today, June 16th, 2008, I'm interviewing Dr. Gerhard Baumann for the Endocrine Society at its annual meeting being held this year at the Moscone Center in San Francisco. Dr. Baumann is an internationally known endocrinologist and expert in growth hormone biology. He has served the Endocrine Society in a variety of capacities, including service on the editorial board of Clinical Endocrinology and Metabolism and Endocrinology, as well as service on the JCEM Pfizer jury. Dr. Baumann discovered and characterized growth hormone binding protein, and currently he is Professor of Medicine Emeritus at Northwestern University. Dr. Baumann, please tell me where your parents were born and what kind of education did they have?
My parents were born in Basel, Switzerland, and they had secondary school education, my mother was a homemaker, my father was an executive in a firm, and their education was not university, but secondary school.
And when and where were you born?
In Basel, Switzerland, 1941.
And was education a priority in your family?
It was a priority, yes. Yeah. But I didn't have to be pushed. I was interested myself.
Mm-hmm. And what kind of education did you have?
I had elementary school, followed by what's called gymnasium, which is 8 years of secondary school preparatory for university education. And it was called a classical gymnasium, which is a school that emphasizes the classics, and was one of the best schools available in the town at the time.
Say that last part.
Was one of the best schools available in Basel at the time. Still is.
Were you making any decisions at this point about what you might want to be doing later in life?
It was at the early phase of my gymnasium time. I was too young to make any such decisions. Later on, the decision certainly was to attend university, but exactly what field was not clear at the time until later, in the later years of my schooling.
Okay. And why did you choose Basel University?
Because it was in the same town, and it was a natural place to go to. Mm-hmm. This would be standard in the European system, at least in the Swiss system, you go to your home university.
And this was in 1960 to 1967 period?
Correct, yes.
Okay. And what was life like socially in that period?
It was, I lived at home. I went to classes, as everybody else did. It's not a campus situation in Europe for the most part. So you went to classes, you had a beer with your colleagues at night, and you came home late or early in the morning. Mm-hmm. And it was not campus life, but similar in a more dispersed fashion.
And how did you envision your life as a future physician at that point?
Well, I decided to study medicine after entering university, because I was interested in science, but I didn't want to necessarily only do science, but have some human contact, and medicine would provide both of these aspects.
And then you did your residency in the United States. How did you decide to do your residency in Brooklyn, New York? Well,
I was interested in coming to the United States, or at least go abroad and look at a different system of education. And I was looking around for open positions in the United States. I picked the cities, and not having any particular guidance on where to go or how to go about it, I simply wrote to various places that I thought might be interesting and for me in terms of cultural environment, and found an open position.
And you were looking at large cities?
Looking at large cities, cities that provided enough excitement for me to do things other than
well,
medicine.
When you came to the United States, that was the late 1960s. How did that
Correct,
strike
68.
you in terms of excitement?
Well, the social upheaval in the United States at the time really was not something that I was particularly concerned about. Perhaps wrongly so. I just didn't know too much about it. And I was basically working in the hospital full time and really had no major contact with the upheaval that was going on at the time.
And what features stand out about this phase of your training?
It's a busy time, learning a lot, taking care of patients day and night, and being in New York and going to when the time allowed, going taking advantage of all the amenities that New York permits.
And how did you picture your life as a physician at this point?
At that point, I had pretty much decided I wanted to pursue an academic career, and I was already interested in endocrinology, and most likely would become an endocrinologist.
And then you got a fellowship at Harvard Medical School?
Right, Peter Van Brigham Hospital.
And what research were you doing there?
I worked in, George Thorne was the endocrinologist at the time, and also the chief of medicine, and he was a major mentor and role model in my life. And there were several labs under his overall guidance. I worked in Joe Dingman's lab, and that lab worked on vasopressin, or antidiuretic hormone. And I learned my first laboratory techniques in that lab.
And then, let's see, you got a position at the NIH in 1974?
Yes. When I went back to Switzerland for 2 years to finish my clinical training, my general internal medicine training, went back to Basel and a year at the University of Lausanne. To finish that for two reasons. Number one, I wasn't sure whether I wanted to stay in the United States at the time. Still sitting on the fence whether that was a good idea or not. I wanted to go home, and I also had a J1 visa, which I had to go home because of that.
Okay. And but when you were at the NIH,
so then after that, I decided it would be better for me for an academic career and for the type of work that I wanted to do, namely a major laboratory career. The environment in the United States would be better. That's when I decided to come back and do some additional training in the sciences, and became a visiting scientist at the NIH at the time.
Is that when you decided to stay in this country?
Yes.
Yes. Was that a difficult decision to make?
It's not easy to leave your home permanently, but what was dominant is the desire to pursue my interests.
And could you say a little bit more about what the advantages were in the United States at that time?
There was more time for academic endeavors as opposed to patient care. At the time in Europe, it was very difficult to as a physician, to have enough time to do laboratory research because of the system that didn't allow that.
And what was going on in your field at the time that you were at the NIH?
Well, in the pituitary field, I was in a lab run by Andreas Grombach, who was working on growth hormone and different growth hormone forms, molecular forms at that time. One of the things that he found is that growth hormone comes in more than one form, and in particular that there's a cleaved form that was more bioactive than what we knew as growth hormone at the time. I happened to be in that lab and pursued that line of work during the time at my NIH and also afterwards.
So Dr. Grombach, you would call him a mentor for
Yes.
you? Yeah.
Mm-hmm.
And what did he teach you?
He taught me a lot of electrophoretic protein chemistry. He taught me protein chemistry, a lot of electrophoretic techniques that allowed the separation of these various forms of growth hormone, and just biochemistry in general, and techniques applied to biochemistry.
What was he like as a personality?
He was a very independent sort, and had his own way of doing things, but was basically a very a freewheeling spirit, and he allowed me to do what I wanted to do.
Okay. And what research were you doing?
Well, I was involved in trying to isolate these various molecular forms of growth hormone, including these more bioactive forms, which in retrospect turned out to be extraction artifacts, but they were interesting extraction artifacts in the sense that they had higher biological activity. So we tried to isolate those, and then once it became clear that perhaps they were not naturally occurring forms, it became important to pursue what are the naturally occurring forms. And that led to the identification of these various native or naturally occurring iso forms of growth hormone.
Okay, and then you went to Northwestern Medical School?
Correct. Mm-hmm.
And what made that position so attractive to you?
Well, I was looking around for a university position at the time, and again, I wanted to be in a major city, and it was a very good environment, a very nice place to be, a very nice campus, great city, and a good position. So that's where I went.
And did you continue with your research on pituitary?
Yes, I continued along the same lines.
And what questions were you asking about?
The main question at the time was what among these various molecular forms of growth hormone which are artifact from extraction from pituitary and which are naturally occurring. So we went about in my lab extracting large amounts of serum, trying to isolate these various circulating forms and finding out what is actually not what has not seen an extraction process, but rather what is natively circulating in blood.
I see. And what were your findings?
And one of the main findings, and disappointing findings to some degree, was that this highly bioactive cleaved form did actually not circulate in blood.
Why is that so?
Well, it's always nice to have a more active molecule than a less active molecule. And I think one of the thinkings at the time was that perhaps growth hormone, as we know it coming from the pituitary, needs to be activated by a process, proteolytic process in the periphery or in the pituitary during the secretory process in making it more active, similar to, for instance, pro-insulin and going to insulin. However, that was not the case, it turns out.
Okay, but how did it advance the field?
Advanced the field in actually making clear that growth hormone is more than one substance, that it comes in these various molecular forms, and these are native forms, and they occur naturally, and it sorted out fact from artifact, basically.
And who supported you in this research?
This was NIH supported.
Okay. Are there further implications of complexity in growth hormone?
The implications of this work were really two major implications. One has to do with the measurement of growth hormone in serum as a diagnostic test. Different labs and different assays give you different numbers. And one of the main reasons for that is the heterogeneity of growth hormone as it circulates in blood, and not every assay or not every antibody or every assay seeing these various forms in the same way. So that's one major implication in the difficulty of coming up with a solid number of what a growth hormone level should be or not. Mm-hmm. The other one is, so that's one. Okay. The other one is implication really for abuse of growth hormone among athletes or bodybuilders. And the molecular heterogeneity that is naturally occurring is not occurring in injected growth hormone, which is a pure single substance. So the implication of this heterogeneity for detecting exogenous or for differentiating exogenous growth hormone from endogenous growth hormone has been used by the anti-doping agencies, or is being used in developing assays to detect growth hormone abuse in athletes. And so that's currently ongoing.
Are you involved in that?
I've been involved with the agencies in consulting, yes.
And where did these questions you were asking about the complexity of growth hormone, where did they lead you next?
Well, one of the side products of this whole research was and one or two unexplained components in blood, which turned out to be the growth hormone binding proteins. The main one was basically an unexplained form of growth hormone in blood that was always hidden away in what's called big growth hormone, or what at the time was called big growth hormone. Now, big growth hormone is a combination of things, polymers, oligomers of growth hormone, but there also was this complex between the growth hormone binding protein and growth hormone, which is one component of the so-called big growth hormone. When we looked at big growth hormone, there was always this component or this peak on columns that we couldn't explain. And one of the thoughts was maybe after all, this is a growth hormone form that is bound to another circulating protein. And to make a long story short, that's what it turned out to be. And hence the discovery of the growth hormone binding protein.
Now, your discovery, it came up against the dominant dogma of the time.
That's correct, yes. Peptide hormones were supposed to be circulating in the unbound form, and that was a pretty strongly held dogma by the endocrine community in general. And one of the, particularly promulgated by one of the Nobel laureates, Rosalind Yalow, who earlier on had very clearly stated relatively weak evidence at the time that growth hormone was bound to a serum protein was almost certainly not the case. And so this was the prevailing dogma at the time.
And what allowed you to see growth hormone binding protein where others had not?
Well, it takes a prepared mind, perhaps, and also the techniques that allow you to separate things. So certainly I had the latter and perhaps the former too. And I had just this unexplained phenomenon of a peak that I couldn't explain, and pursued it as to its nature, and that's what came out.
Could you outline the critical experiments?
Well, one of the critical experiments was to postulate maybe there is such a binding phenomenon, and let's show whether we can bind growth hormone to serum proteins. So it's actually a relatively simple experiment which had been done before in a more crude way by others. You radiolabel growth hormone, incubate it with serum, and then you separate it on a column and see whether it has the same size as before. And what happened is that the smaller size of growth hormone was converted to a bigger size after incubation with serum. Now, this could be a number of different things, including aggregation, which everybody had thought that's what it must be. Mm-hmm. But then we went on and actually purified this component, and showed that it's composed of both growth hormone and the growth hormone binding protein. Mm-hmm. And there's actually yet another series of growth hormone binding proteins that are even bigger. So there's also more than one growth hormone binding protein.
Okay. How difficult was it for you to get your findings published, and what was the reaction in the endocrinology community at large?
Well, the publication wasn't that difficult. I think one reviewer rejected it just based on the dogma, and the other two reviewers couldn't really ignore the evidence completely, so it was published. But there was a lot of skepticism for a number of years whether this was actually a true finding. What helped is that other workers or colleagues, namely Adrian Harrington in Australia, had found the same thing, and also described the same binding protein in his lab. And there's yet a third one who was involved in the field, Mike Waters, who also had evidence to the same effect. So I wasn't alone in fighting this battle.
Did the reaction that you got surprise you at all?
Not necessarily. I think I knew I was up against a strong dogma. Mm-hmm. So I wasn't surprised.
How did you feel confronting a strong dogma?
Well, it's somewhat intimidating, perhaps, but it's also fun to debunk existing dogmas. So it was. I think the evidence was so strong I couldn't ignore it, and it certainly helped me being recognized for somebody who doesn't back down from a fight.
How did growth hormone binding protein eventually advance the field?
Well, the growth hormone binding protein turned out to be a fragment of the growth hormone receptor, namely the extracellular domain that's shed from cells and floats off into the circulation and circulates as a soluble receptor fragment, which contained the binding site for the hormone. And this circulating receptor was relatively easy to sample and measure, as opposed to the full receptor, which is on cells, and you can't really sample it certainly in humans very easily. And it gave us a window on receptor biology, or growth hormone receptor biology, and growth hormone receptor physiology, how it's regulated, and how it's the biology of how it functions in various physiological and pathophysiological states. So it provided an assay or a measurement of growth hormone receptor abundance, perhaps, and perhaps function that would otherwise not have been possible as easily.
What are the diagnostic implications of that?
The only really established diagnostic implication is, or use, I should say, diagnostic use, is in a disease called Laurent dwarfism, which is a mutation in the growth hormone receptor. Most of those patients can be diagnosed, other than clinically. The critical diagnostic step would be the absence of the binding protein in blood, because the receptor is absent, as is, therefore the binding protein is also absent or dysfunctional. It's not true for all patients, but for the vast majority. That's one implication. The other implication is the binding protein complicates, once again, the measurement of growth hormone. And you have to basically, in the assay, taking into account the presence of the binding protein and its abundance or its concentration and its possible interference in the assay.
Does growth hormone binding protein enhance growth hormone activity?
It can. It does two things. In vitro, it's clearly inhibitory. It competes with the growth hormone receptor for binding of the hormone. So it's therefore inhibitory. It also probably acts as what's called a dominant negative player, in the sense that it forms heterodimers with the receptor, and the receptor can only signal as a homodimer. And if there is exchange of one receptor versus a binding protein in this dimer, that will be a dysfunctional receptor and or a dysfunctional complex that cannot signal. So it will be inhibitory along those lines also. However, it also prolongs the half-life of growth hormone in serum. So there's some experiments showing that if you give enough binding protein, you can actually enhance, in vivo, in a rat, you can actually enhance the effect of growth hormone, probably via prolongation of its half-life. So it's a complicated situation with both local inhibitory or negative regulatory effects and more global enhancing effects. Exactly where that balance lies in physiology is still not known.
Who are your collaborators on all this?
Well, I had several fellows in the lab, and we were pretty much alone at the time. There was Adrian Harrington's and Mike Waters' labs were also working on this, but we were pretty much working independently. So the collaboration was local, not outside.
Okay. Are there any implications of growth hormone binding protein beyond endocrinology?
Well, it was the first example of a circulating, I believe it was the first example of a circulating ectodomain of a membrane receptor of the cytokine family, cytokine receptor family. And since it's been shown that most of these cytokine receptor members have similar cleaved or circulating ectodomains. So it was an example of a much bigger phenomenon.
So the new tool that you had basically with growth hormone binding protein, this was in the mid-1980s, is that correct?
Correct, yes.
Okay. Why did you then study the levels of growth hormone binding protein in the African pygmy?
Well, we were looking for a number of physiological states where there might be abnormalities in growth hormone receptor biology or function. And the Laurent dwarf or the Laurent syndrome patient would be a classical example of somebody who there's a genetic defect in the receptor and therefore a genetic defect in the binding protein. So this was the first example. But we were looking at all kinds of short stature and tall stature syndromes, looking for either impaired or enhanced growth hormone action via the receptor. So the pygmy was just one of those unexplained short stature phenomena that we pursued in collaboration with Dr. Mary Mee, who had sera stored from pygmies in Africa. And that was a collaboration with him in terms of finding out whether the pygmies actually had perhaps growth hormone resistance based on impaired growth hormone receptor biology. And they do have low growth hormone binding protein, so they may have abnormal growth hormone receptor. Although there are other things in pygmies that also occur and are not explained by this finding. So it's still not clear exactly why the pygmies are short. Nutritional effects, growth hormone resistance effects, maybe IGF-1 resistance effects. So it may be multifactorial.
I see. How did you find yourself on a desert expedition to Pakistan in the late 1990s?
Well, that's an interesting story and a fun story to tell, actually. There was this, one day I received a letter from Karachi, Pakistan, by one Hiralal Maheshwari, MD, PhD, who said, who enclosed in the letter a newspaper article entitled "The Dwarfs of Sindh." Now, Sindh is the southernmost province of Pakistan, and the article described a village, or two villages actually, where there was a cluster of very short people, and they were all related, and they were all men. And it sounded like it was either a genetic problem or a local problem in the water or whatever. And he asked me, would you like to study this? And so it sounded interesting to me. He included a crude pedigree, and it looked like there might be some recessive genetic trait involved here. So I wrote back and said, well, go and see if it's true. Go to the village and see if it's true. And if you're there, see if you can get some blood and send it here, which he did. And he confirmed it, and he actually came himself with the blood samples. And that's how the long story started, and trying to find out why are these people so short, and what's the genetic underpinning.
And let's see, who funded this expedition?
So then we found out from the blood samples he brought that they probably had growth hormone deficiency. So we knew that much. We knew it was probably a pituitary problem, but we didn't know which one. So we said, well, we have to go back and get DNA and RNA and study them properly with dynamic tests and so on. So we organized an expedition to go to the village, or the two villages, and do all that. Who funded it? It was funded, the expedition itself was funded by a Northwestern intramural grant.
Okay. And let's see, what questions were you asking?
So the question then was, well, it looked clearly genetic, and it became very clear this is a recessively inherited disease. And they're all short because there's consanguineous marriages, and only those from consanguineous marriages were actually short. Turns out most of the marriages in those villages are consanguineous at some level, a high degree of inbreeding. So we went there and we studied. We did pituitary function tests. We measured their height and BMI and everything. Looked at their clinical phenotype in detail. We got DNA, we got RNA, we got cells, and we brought all this back to my lab. And again, to make a long story short, we found a mutation in the growth hormone releasing hormone receptor. Actually, the first such mutation in humans. A mouse analog had already been published of that, and it was a new syndrome that had not been previously described, except a few months before our paper. Another paper came out from New York describing the same mutation in 2 patients who were short and of Pakistani descent in New York.
Wow.
So we weren't quite the first, but almost.
When you were doing your work there, how did the local population react to you?
Well, at first they were very skeptical. You don't want to come as a colonialist and and use these people. But thanks to my colleague Gerhard Baumann, who was a local, we overcame this very quickly, and we became very, very cooperative and very friendly. So it wasn't really difficult on the human level. I think the society and the mullahs and the religious leaders were still a bit skeptical, but that was overcome.
What solutions, either therapeutic or social, resulted?
Well, these patients, of course, they have a null mutation in the growth hormone releasing hormone receptor, which means the brain cannot signal to the pituitary to either make growth hormone, to make enough cells that produce growth hormone, or to release growth hormone from the cells that store it. So therefore they should respond to growth hormone, and indeed they do. So if you give them exogenous growth hormone, they respond perfectly normally. That's not, and we tried that. It's not easy to do in Pakistan, at least. We were not unable to import growth hormone to Pakistan and have a local physician actually take care of these patients and do it properly. So the sad story is they're still not being treated in Pakistan. However, there are many other such mutations. Since then, there have been many other such mutations described in different parts of the world. A very big cluster is in Brazil, northeastern Brazil, town Itabaianinha, with a similar mutation in the same gene, also inactivating, and the same phenotype, and they're being treated. And so it's first of all, he put this new syndrome on a map so that people could start looking for it. So many more patients have been found since, and for the majority is being treated with growth hormone.
Are there any social solutions being tried in Brazil? Can they be applied there?
Not sure whether that's, they certainly could. What you want to avoid is consanguinity. That's easily said and not always so easily done in certain societies. And we found that out in Pakistan. They understood perfectly what the problem was after we explained it, but society was not ready to accept a different mode of marriages and so on. So I don't know what the future will bring.
Okay, I'd like to ask you a little bit about the Endocrine Society now. What are some of the boards or committees that you've served on?
Been on the membership committee. I'm currently been on the JCM Pfizer jury, currently the chair of that. I've been on the editorial boards of endocrinology and JCM for a total of 8 years probably each. And I do many ad hoc reviews for journals of the Endocrine Society.
And why have you chosen service on those particular boards?
I was asked to join those various boards, so that's why I did it.
And in what ways has the Endocrine Society supported your personal development, your professional development?
The Endocrine Society is really the home, has been my home scientifically and also clinically, as a place where you can present your work, where you meet your colleagues, and where there's a high standard of science and also a high standard of patient care. So it's been the main scientific home for me.
And what are your current views of the field?
Field of endocrinology
Yes.
in general? Well, it's still an exciting field, and it's a very interesting cerebral specialty that's very poorly paid for. You're not paid well for thinking, for the most part, and so there's some concern about the endocrinologist being, at least the clinical endocrinologist, being a vanishing species because reimbursement is very poor, and now funding for scientific work is also very difficult. So there's some pressure on endocrinology currently, but hopefully we'll overcome that.
Okay. Okay. Did you want to say any more about the issues?
No, I think we pretty much
Okay.
covered
Great.
everything you wanted to hear.
Well, thank you.
Okay, thank you very much.